consensus sequences in prokaryotes. So that's all you need to know in terms of prokaryotic binding, is that the sigma subunit of RNA polymerase is looking for a consensus sequence to bind to. I believe it is looking for the -35 region consensus sequence, and DNA will open up at the PribNow box. So, oh yeah, here's the vocab term, PribNow box, which is the consensus sequence that you find in prokaryotes. And here's the reason why it's often confused with Tata box. Look at the pattern of the PribNow box. That's why it is very often confused with Tata box in eukaryotes. Yes?
So DNA is originally attached, but it scans and then it stops at the -35, right? Once it recognizes it? Yeah,
so the scanning thing, it's more like it's binding and dissociating from a bunch of random areas in DNA. So yeah, you can think of it as scanning.
Does it bind with low affinity at first?
I'll get to that when we get to initiation in a second. So, before we get to the steps, this is very, very important. You will get 1 or 2 questions on this following concept here, is consensus sequences. You may be given multiple different strands of DNA, multiple different promoters, and you may be given a consensus sequence. And you may be asked, which one is the strongest promoter? We talked about that at the very beginning. What's the first thing you need to look for to see if something's a strong promoter? It looks similar to the consensus sequence. The more exact it is to the consensus sequence, the stronger the promoter. Now, what if you have multiple answer choices that look exactly the same as the consensus sequence? You break the tie by looking at the other places around the consensus sequence. The ones with more A's and T's in it is better, because what's the purpose of these promoter sequences? It's where DNA is going to open up so that you can transcribe it. So, once you have your promoter, once you have your consensus sequence that looks really good, if you want to break some ties between consensus sequences, look at everywhere else. How many A's and T's do you have there? If there's a lot of A's and T's, it's easier to open up. That's a good promoter if it's easier to open up. Yes?
Will the consensus sequence, will it be given to you?
Yes, they will give you the consensus sequence, so don't worry about memorizing it. Yes?
Does the promoter be the sigma subunit?
The sigma subunit is let me get a picture for you here. The sigma subunit is on RNA polymerase, so it's actually part of the enzyme, and it will bind to the consensus to your consensus sequences on the actual DNA. Yes.
What is the helix term helix on that part?
So, it is important probably for 29B, in other words for the final exam, but this is what's called a DNA binding domain, or a DNA binding motif. I'll go over it more in detail when we're reviewing for the final exam, but a short answer is that any protein that has a DNA binding domain has the ability to regulate transcription, because it binds to DNA and does something to it. Other questions regarding consensus sequences? All right. Now the actual steps of transcription. I will start with this diagram here. It's a very good diagram describing initiation. It's a very well illustrated diagram. So, transcription is separated into 3 steps: initiation, elongation, termination. The initiation step involves the actual binding of RNA polymerase to the target DNA that needs to be transcribed. It will do so by binding at the promoter region via the sigma subunit. So let's follow the diagram. We see that it binds here, and that binding is called a closed promoter complex. Here's why it's called a closed promoter complex. Notice that the DNA strand is still closed. This means transcription has, the enzyme has not committed to transcription yet. That means this binding here is actually fairly weak. It is relatively weak because it hasn't decided if it wants to commit to transcription yet or not. So imagine this thing is doing this at several different regions in the genome. It's looking for a good promoter sequence. It's looking for a gene that it wants to transcribe, and it does so by initially forming a closed promoter complex. If it is going to commit to transcription, it will have to open up DNA at the PribNow box region. When it does so, you form the open promoter complex. This RNA polymerase is tightly bound to your DNA now, and it has committed to performing transcription. In fact, in the initiation phase, it will synthesize the first few nucleotides in your mRNA. The very first one will always be a purine. That is a unique feature of prokaryotic transcription of RNA polymerase, is that the very first base is going to be a purine, which are what 2 options? Anyone remember? A's and G's. So your first nucleotide in your mRNA will be either an A or a G because of the initiation phase. Now, in order for initiation to end, after a few other nucleotides are added, we have a problem. That sigma subunit was there to force RNA polymerase to bind to its template DNA. However, in order for RNA polymerase to transcribe the rest of this gene, the last step is the sigma subunit needs to leave. The sigma subunit dissociates, you remove the parking brake, RNA polymerase will transcribe the rest of this gene, which signifies the end of initiation, is sigma subunit leaving. Yes?
When does the binding come in? Like, is that at the transcription start site, or?
That's a good question. Very, very important. Let me go back to that consensus sequence picture. The binding of RNA polymerase does not occur at the initiation site, does not occur at the transcription start site. The binding occurs what the term we use is upstream to the transcription start site. You see these negative numbers here? In this class, at any time in the future, if you see negative numbers associated with the DNA sequence, that means it is upstream to a transcription start site. Okay? If you see positive numbers, we'll see more of that in 29b, that is downstream of a transcription start site. So, binding happens upstream of the transcription start site. Okay. Whoops. So some of the information I described with initiation is actually described here, but there's some extra bit of information added on this slide, such as it turns out we have a whole set of antibiotics that specifically inhibit prokaryotic RNA polymerase. That's great. That's one way we can kill bacteria is by inhibiting the RNA polymerase in prokaryotes. And this is good because these drugs are specific enough that they will only target prokaryotic RNA polymerases, and therefore the eukaryotic stuff can work just fine. Yes?
Do you have to know the role of each, like the specific role?
No, just general. If you see these terms on any answer choice or question, make sure you know that they're there to inhibit prokaryotic RNA polymerase. Okay? A lot of this we are, oh, no, this is elongation. Okay. So, after initiation, which is the sigma subunit leaving, you now have elongation. Elongation is actually really simple. It is the actual transcription part. You're adding nucleotides to an extending RNA strand. You always add nucleotides to the 3' end of your extended RNA strand. One feature to worry about when we talk about eukaryotic transcription is this issue with supercoiling again. In order to transcribe DNA, you have to open up DNA. And whenever you open up DNA, you build supercoiling. You create supercoiling. Turns out, ahead of this transcription bubble, you create positive supercoiling, and behind it, you leave behind negative supercoiling. We have a set of enzymes to fix both of these things. Gyrase will fix positive supercoiling, and general topoisomerases will fix negative supercoiling. And that's all you need to know about elongation. Let's see. There are 2 ways to end transcription. This is the end of prokaryotic transcription, and that is row-dependent termination. And although it's called specific sequences here, another way to describe this is row-independent termination. And let's illustrate both of these. This diagram here is a very good illustration of row-dependent termination. So, what we have here is that mRNA is being made through transcription, and there's a protein called Rho. So this only is for row-dependent termination. There's a protein called Rho that will bind to mRNA at a specific site called the RUT site. And what this Rho protein will do is it'll travel along the mRNA in a 5' to 3' direction until it encounters the RNA polymerase. Now, it'll create this hairpin loop, but that's not the important part here. When it catches up to this RNA polymerase, it will use, the important part, the helicase activity to unwind everything from each other on this RNA polymerase. It'll unwind the mRNA from the DNA, and if you do that, the RNA polymerase loses the ability to continue transcription at this particular gene. So that is how row-dependent termination works, is through the activity of Rho. Yes?
So Rho binds to the RUT how? Through a consensus sequence?
Yep, a consensus sequence at which you don't have to memorize either. But yes, that is exactly right. The RUT site contains a consensus sequence for the Rho protein. So we can contrast that with row-independent termination, which doesn't involve any proteins. It actually is just the sequence itself. Remember when we were talking about cruciforms and how we have to find palindromic sequences? It turns out that when you make mRNA, and if that mRNA was made from a palindromic sequence in DNA, a synonym is inverted repeat, if that was a palindromic sequence that ended up being transcribed, that mRNA sequence will end up binding to itself to form this structure here called a hairpin loop. A hairpin loop visually looks like a cruciform, right? It's just half of a cruciform. A cruciform is that cross shape. mRNA, when it forms this, when it's binding to itself, it forms a hairpin loop. This hairpin loop, it turns out, is actually very unstable for the RNA polymerase. In fact, there are two features to end transcription via row-independent termination. The formation of a hairpin loop, and notice on the tail end you have a long string of uracils. That's a very weak type of base, right? Because if this was bound to DNA during transcription, it's binding to those A's in DNA, and we already know that AAT binding is very weak. Same rules with AU binding. A's to U's is a very weak association. So when you form a hairpin loop, which is already destabilizing the RNA polymerase, when the tail end of this hairpin loop is a bunch of uracils, it's a very weak binding, so quite literally the mRNA is just going to fall off the template DNA, and you end transcription without the need of a row protein. That is row-independent termination. All right? And there's additional pictures to describe that, which I will skip. I don't remember exactly where they say the following, but when you compare RNA polymerase to your DNA polymerases, how do you compare its accuracy? Well, DNA polymerases have mechanisms to fix errors, right? Those proofreading mechanism, editing mechanism, stuff like that. RNA polymerases don't have that, so they are more susceptible to errors. In transcription, it might accidentally put in the wrong base at the wrong place, but that's fine. Evolutionarily speaking, there isn't usually a lot of consequences in terms of messing up transcription, because you can always transcribe the gene again and it might be correct the second time you do it. So RNA polymerases do not have that repair mechanism, so it's more susceptible to errors. Yes?
Well, that's good because then they can adapt, right?
Adaptation actually is at the DNA level. So at the RNA level, you don't really have a lot of consequences for messing up and having mutations at the RNA level. Mutations at the DNA level happen when DNA polymerases are unable to fix errors in DNA. So, oh yes, regulation of transcription in prokaryotes. And I bet a lot of you are probably really interested in this one. So, I am tired and we're running out of time, so I may not go over it in as much detail as I usually do, but luckily I've gone over this extensively at SI, so you can review that video for more information. But we are going to talk about the Lac Operon. Regulation of transcription in prokaryotes. So, a good example of regulating transcription in prokaryotes is via something called the Lac Operon. This is an example of regulating transcription in prokaryotes. The way the Lac Operon works is that, remember in prokaryotes, genes are contiguous, they're right next to each other. Well, it turns out in the Lac Operon, you have a region called an operator, and you have a string of genes where collectively you call this an operon. It is the Lac Operon, an operator plus a set of genes that are the component of, well, let me just go ahead and write out a couple of genes. I don't remember the exact names, so forgive me if I just put A, B, C, D here, and you don't need to know the specific names of the genes. But the Lac Operon is composed of these sets of genes that when you put them together, when you transcribe and translate these into functional proteins, the important part is that the Lac Operon makes enzymes that metabolize lactose. Okay, they will metabolize lactose. And you really only want to do this if you're lacking in glucose. All organisms prefer to metabolize glucose because it's easier and you get more energy out of it. However, sometimes if you're in a situation where there isn't a lot of glucose, and bacteria have found a way to metabolize lactose as kind of a backup plan. So, in order for this to happen, the Lac Operon needs to be activated. And this happens in a two-step process for full activation. And I'll go over the two-step process first. One, you turn on the Lac Operon via a mechanism called negative inducible. And at SI I go over the 4 combinations of these terms. For the Lac Operon, I'm only going to go over the 2 important ones. Negative inducible, what this means is the negative part means a repressor enzyme or repressor protein is involved in some way. Repressor protein involved. The inducible part means your goal here is to turn on or boost, increase transcription. All right? So how do these two terms work together? It sounds like they're opposite things. Here's how it works. The only way for these two terms to make sense is if initially, and you know it's a good sign if a lot of you are bored about me talking about this, it means you know this very well, so that's good. So you have the operon here. The only way these two terms put together make sense is if there's a repressor that is by default on the Lac Operon.
Does it have to be on the operator or in the Lac Operon?
Yes, the repressor will bind to the operator site on the Lac Operon.
Okay,
yeah.
not the Lac, okay.
Yeah. And in doing so, it's preventing RNA polymerase from coming in and transcribing the genes of the Lac Operon. Okay. So the repressor binds to this thing. Now, the inducible part means if you want to turn on the Lac Operon and activate transcription, you must remove the repressor. But what will actually do that? Well, there are molecules, smaller molecules called co-activators, or co-inducers, excuse me, co-inducers, which function to bind to the repressor, cause the repressor to change shape slightly, and it'll just fall off the Lac Operon. Okay? The co-inducers you need to know, the co-inducers are going to be lactose. And anything that is a kind of similar to lactose structurally, such as allolactose. I am oversimplifying it, but this is enough information for you guys in terms of this co-inducer stuff for the Lac Operon. Yes?
In the PowerPoint, you referred to something as like a metabolite. Was that like the co-inducer binds to the metabolite, which is the repressor?
Yeah, metabolite is kind of, and that is important to know, is that metabolites, whenever you see that phrase, refers to these little molecules like co-inducers, co-repressors, and byproducts of metabolism that affect the regulation of certain genes.
So the metabolites are the co-inducers?
Yes, metabolites is an umbrella term. Co-inducers is the specific thing. All right. So, what is the end result of negative inducible? You remove the repressor via these co-inducers, and you are left behind with the open Lac Operon. All right, that's step 1. In order for full, complete transcription to happen, you must do step 2. And I will explain all the different versions that you got very loosely, but then you guys will need to give me more details for the extra credit if this happens to be your extra credit question. Step 2 is now that you have, the repressor is gone now, step 2 involves a second thing that works via positive inducible. And the phrase positive inducible means, positive means an inducer protein is involved in some way. The phrase inducible is still the same definition as it was earlier. Your goal is to turn on or increase transcription. All right? And in fact, for step 2, there is a specific name for the inducer that you need to know. Anyone remember what it's called? Catabolite activator protein, the CAP protein. So that is the actual name of the inducer. This is step 2 in terms of regulating the Lac Operon, And this is initially not bound to the Lac Operon. You have to make it bind to the Lac Operon via co-inducers. But these are different ones. Co-inducers. Anyone know what the co-inducers are here? Exactly, cyclic AMP. These are the co-inducers for the cap protein. Yes?
So where does the cap protein bind? Does it bind to like the cap site, or does it bind to the operator?
It does bind to the cap site. I'm not really familiar with exactly where, but as long as you describe that its job is to bind and turn on transcription, you're fine.
Where does it bind? For a multiple choice question, where would it bind?
Oh, cap site, definitely, yeah. So the cap protein binds to a named site called the cap site, which is somewhere on the operon. You don't have to illustrate that for me. But as long as you indicate that the presence of a co-inducer, and you need to tell me what co-inducer, cyclic AMP, if it binds to the inducer, it changes the shape of the inducer a little bit so that it now is able to bind to the Lac Operon. In doing so, you really increase transcription to its full extent. So you have full transcription.
So the cap binds to the operator. Correct?
It binds to somewhere on the Lac Operon. You can use the word operator, you can use the word cap site, either one is fine. But it binds to this to turn it on, yeah.
So if there's no glucose and a lot of lactose, is it also full transcription? Or
That's the next thing that we're going to go over is the different states that you have. But this is the general features first. It's very important that you tell me the general features first. The step 1 and step 2, yes?
Do the co-inducers bind to the cap protein before the cap protein binds to the Lac Operon?
Exactly. The cap protein is unable to bind to the Lac Operon unless the co-inducers bind to the cap protein first. Exactly. Other questions? Okay. So, step 1 is removing the parking brake, removing the repressor. Step 2 is stepping on the gas, adding the cap protein to the Lac Operon so that you fully transcribe this gene. Yes?
So 1 and 2, those are part of the Lac Operon, how it modifies the transcription.
Yep. Both are necessary for full regulation of the Lac Operon. However, I should mention the following. If, in a multiple choice question, they ask, how is the Lac Operon regulated? Technically, it is just this one. Okay? If a multiple choice question is asking you how the Lac Operon is regulated, technically it is only this first part. However, in an extra credit question where I want the full picture, all the steps that could possibly be involved in the Lac Operon, I want both step 1 and step 2 described. Okay? So, the last part of this, to round out our explanation and detailing of this, is to tell me what happens in the 4 possible conditions. And I might put this in non-logical order here. You don't have a lot of glucose, and you have a lot of lactose. When you have a lot of glucose and not a lot of lactose. You don't have a lot of either of them. And you have a lot of both. Okay. And actually for these parts, I want you to tell me what happens to step 1, what happens to step 2, and what is the end result for each of these combinations here. Let's break it down. I'm going to simplify it, but you need to explain it to me. For step 1, if you have a lot of lactose, what happens to the repressor? It comes off, right? Because lactose is the co-inducer that helps the repressor be removed in step 1. What about glucose? Now, you can add it to this diagram, and I did forget to do that because I was waiting for here. But how does glucose have anything to do with step 2? It turns out that the amount of glucose is inversely proportional, which is indicated with this sign here, this alpha sign here, is inversely proportional to the amount of cyclic AMP. So we know cyclic AMP is the co-inducer for the cap protein. So if there isn't a lot of glucose in the cell, for various reasons that take a long time to explain and you don't need to know, there's a lot of cyclic AMP. Okay, so note that they are inversely proportional to each other. So if there isn't a lot of glucose here, you need to tell me there's lots of cyclic AMP. And if there's lots of cyclic AMP, tell me what happens to the cap protein in this case. All right, I'm not going to go over in detail because you need to tell that to me. You need to describe in detail what's going on for this first scenario. Second scenario, you don't have a lot of lactose. The repressor in step 1, therefore, cannot be removed. And if you can't remove the repressor, honestly, it doesn't matter how much glucose you have, because if you can't remove the repressor, you cannot conduct transcription. So there isn't transcription in either of these two cases. Yes? Yes, that's the key thing, and that's what a lot of people forget, is that this is a two-step process, not happening simultaneously. The first step must happen in order for you to have full transcription. The first step is the removal of the repressor, which can only happen in the presence of lactose. So if you don't have any lactose, it doesn't matter what happens to glucose, you cannot perform transcription. Yes?
So for 3, since there's less glucose, there's more cyclic AMP. So,
Yep.
so does CAP technically bind, it just doesn't have an effect?
Good question. Yes, CAP would like to bind there,
But
but
it can't?
it can't because the repressor is in the way. Exactly. Without a lot of lactose, the repressor is there. It would be like you have the parking brake on and you try to step on the gas. That is not a good thing, and you will not be able to fully activate, you will not be able to transcribe the Lac Operon in that scenario. But what happens if you have this scenario down here, if you have both glucose and lactose here? Well, evolutionarily speaking, prokaryotes prefer to use glucose. So what has happened to the Lac Operon? The presence of lactose allows the repressor to be removed. But the presence of glucose means there isn't a lot of cyclic AMP, so you can't actually put, so the cap protein is unable to bind to the operon. The analogy for this one is you've removed the parking brake, but you don't step on the gas. So what term is used to describe this last one? Leaky transcription, which is a little bit of transcription.
So because the cap cannot bind to the Lac Operon, it will cause leaky transcription.
Yep,
and
exactly.
therefore, when transcription is turned on, it will be turned off, but it will be very slow.
It'll be very slow transcription. Yes, that is what leakage is. Yep, it'll be. That is what slow transcription is. Is you've removed the parking brake, so you can transcribe it a little bit, but you aren't going at its optimum pace, which I did forget to write down. That's this one, full transcription. All right. I have not written everything that you need to know for this, so please review it and practice by writing out as much as you can from memory. And like I said, same thing for exam 2, extra credit. Tell me everything you know in as good of a detail as possible, enough to convince me that you haven't cheated by standing in line and copying the person that's next to you, because I know that happens. Okay, so if you want to leave, you can, but I need to go and check to make sure I haven't forgotten anything. So. Oh, well, now let me check this then. Yep, that's it for now. All right, that's it. So I will see you guys if you're planning on sitting inside. Oh, thank you.